Hello everyone, in this video I'm going to speak about real-time PCR. Of course there was a previous video about the basic principle of PCR. So if you don't know anything about PCR, I highly recommend you to go there, watch the previous video about PCR and then come back because then it will be much easier for you to understand this video. The link of the PCR video is in the description of this video, so you can go there directly. Now in this video I'm going to speak about real-time PCR, the principle, the technique, how does it go, and everything related. I also will speak about reverse transcription because it's used widely in real-time PCR, and also the data of real-time PCR. So let's, as usual, let's start by asking what is real-time PCR. Real-time PCR is basically a modified technique from PCR. So what's the difference? As I told you in the previous video, PCR is a thermocycler. So it performs a thermocycle by heating up and cooling down the temperature in 3 steps, which are the DNA naturation, then primers annealing, and then the DNA extension, which these 3 steps represent one PCR cycle. And then after each cycle, we will get double the number of PCR sequence we have. So if you have first 2 sequences, then we will get 4, then 8, and so on. So it's a process of DNA amplifying, an amplifying of a specific DNA sequence or gene. Now, of course, in PCR, what we can do is to set the time. We can set the time for 1 hour, half an hour, or so on. And at the end of the PCR process, we can, of course, we can get our sample and we can measure the amount of DNA we have at the end of the PCR. But we will never know how much PCR we had during the cycle. And this is the modification in the real-time PCR, because real-time PCR is a combination between PCR and spectrophlorimetry. So it's like normal PCR, but it's engaged with spectrophlorometer, which can measure the amount of DNA in my sample continuously during the PCR process or after each cycle. So I will get an estimation how much DNA I had in each cycle of my PCR process. So let's say this is the PCR. We have double numbers of DNA sequences after each cycle. The spectrophlorometer will be measuring the amount of DNA in my sample during the PCR continuously. And this is how does it work? So this is a diagram of the real-time PCR machine. So here is what we call the tube patch. It looks like this from outside. So here is when we put our tubes. Normally we use plates because we can apply real-time PCR to several samples at one time. So we can use the 96 Wells plate or whatever. So here is the thermocycler, like the normal PCR. This what you're seeing here is the normal thermocycler, and what you're seeing here is the extra, which is the spectrophlorometer. So the thermocycler undergoes the normal thermocycle, the 3 steps thermocycle, and then the spectrophlorometer measures. So this is how the spectrophlorometer works. So it excite a specific fluorophore inside the sample, and then the fluorophore emits fluorescent or emits light, and we then detect the light coming out of the sample. So here is how the spectrophlorometer works. So the spectrophlorometer keeps measuring the amount of DNA in my sample continuously during the PCR process. Because of this, it's called real time. Here is how the spectrophlorometer works, basically. So this is the light source. Then we have mirror. We have the monochromator to choose like a specific wavelength. Then the light causes an excitation to the fluorophore, which emits light, and we can detect in the detector. So this is the spectrofluorometer, which we see here, and this is how it works. So this is the real-time PCR actually. In real-time PCR, it's like normal PCR, but we are measuring the amount of DNA we have inside the sample continuously during the PCR. Because of this, it's called real-time. Now I'm going to speak about how does it work? How does real-time PCR works? As I told you previously in the PCR video, that we need 4 essential elements in order to perform a PCR process, which is first we need the parent DNA, we need the DNA sequence or the specific gene we want to amplify. Then we need the DNA contents because we need to synthesize DNA. So we need the DNTPs, which are the deoxyribose nucleotide 3-phosphate, the adenine, thymine, cytosine, guanine, the DNA content. We also need primer. We need a couple of primers, the reverse and the forward. This I told you in the previous video. And of course we need the very important enzyme, the DNA polymerase, the heat-resistant DNA polymerase to produce DNA. Now in real-time PCR, we need one extra molecule, which is very important, and which is the detector. The detector is the thing we use to detect. The detector is what the spectrofluorometer detects. So this is the very important thing in the real-time PCR, the detector. So I'm going to speak about the detectors used in the real-time PCR. We have 2 types of detectors, the non-specific ones and the specific detectors. The non-specific detectors are fluorescent dyes. The most known of them is the cybergreen. The cybergreen is a molecule, is a fluorescent dye. It looks like this, and I'm going to speak about it in details. The specific detection is a detective probe. It's a probe which is very specific to the gene or the specific sequence I want to amplify. The most known is the TACMAN, and it looks like this. It's a probe, and it's very specific to the gene or DNA sequence I want to amplify. So I'm going also to explain this in details. So let's first speak about cybergreen. Cybergreen it's a green fluorescent, of course, fluorophore. From its name, it's green, and it can so it can realize or it can detect the double-stranded DNA. It's very sensitive to double-stranded DNA. So let's say we have a double-stranded DNA. The cybergreen can bind to the minor groove of the double-stranded DNA. So the cybergreen um, um, can sensitively detect the double-stranded DNA, and then it goes to the minor groove and it binds there. Once the cybergreen is bound to the double-stranded DNA, it becomes fluorescent. Okay. So the cybergreen can absorb light at 497 nanometers and emits light at 520 nanometers. But the thing is that cybergreen is not specific. It's sensitive to double-stranded DNA in general, but it's not specific to a certain gene or to a certain sequence. So it can detect any double-stranded DNA in the medium. So let's say this is our sequence, or this is our DNA, and we're applying the PCR. So in the PCR, the sequence will be doubling after each cycle, as I told you previously. And let's say we have cybergreen in the medium. Then the cybergreen will detect the minor groove of the double-stranded DNA and will bind there. So at each cycle, and when the cybergreen is bound to the DNA, it becomes fluorescent. So at each cycle, we will get more fluorescence because more cybergreen will be bound to the DNA. At the end, we will get more and more fluorescence during the PCR process. And of course, the spectrofluorometer can detect this fluorescence. Yeah, the fluorescence will be coming out of the sample, and then the spectrofluorometer can detect the green color or the green fluorescence coming out of the sample. Let's talk about the advantages and disadvantages of cybergreen. So the advantages, cybergreen is easy to produce. It's a cheap product in comparison with other detectors. It's easy to handle, it's sensitive, it's very sensitive and effective to, it's very sensitive to double-stranded DNA. The only disadvantage is that cybergreen is non-specific, and so if, let's say, if there was a mistake with the primers, so if the primers don't bind specifically to the gene of interest, and so another sequence would be produced in the sample, the cybergreen cannot detect this because the cybergreen doesn't differentiate between our sequence of interest and other sequences. So we can overcome this problem by using highly specific primers. Then we can ensure that the primers will bind specifically to the gene or to the sequence of interest. Okay. Now the other type of detectors are the specific detectors, which are the detective probes. And as I told you, the most known is the TACMAN. How do these detectors work? So this detector is a probe. It's a DNA probe. It's a single-stranded DNA, and it's coupled with two molecules. The first is the fluorophore. So first we have the fluorophore at the 5' end, and we have at the 3' end something we call the Kensher. What is the Kensher? The Kensher is an inhibitor of the fluorophore. Why? Because the Kensher absorbs any fluorescence coming out of the fluorophore. So as long as the fluorophore and the Kensher are bound together or are kept closely around each other, the fluorophore cannot we cannot see any fluorescence coming out of the fluorophore because the Kensher absorbs all the fluorescence coming out of the fluorophore. Now, how does this work? How can we detect the amount of DNA in our sample using this? So let's say we have our double-stranded DNA, and as you know, so let's say we have our double-stranded DNA. We want to perform PCR. So the first step of the thermocycle is the denaturation, in which we increase the temperature. So the hydrogen bonds between the nucleotides will be denaturated, and then the two strands of DNA will be separated from each other. So this is the first step of PCR. At this step, the Kensher will recognize, or not the Kensher, sorry, the TACMAN will recognize the strand, our DNA strand, because it's specific. So this is exactly complementary to our gene or our strand or our sequence of interest. So this will recognize the sequence, and then the detector or the probe will bind to our strand, to our DNA strand or to our DNA sequence, because this is specific. The probe is directly complementary to the DNA, to our DNA, so it will bind to it. Now, in the second step of the thermocycle, which is the annealing step, the primers will be binding at the 3' end of the gene. This is also what I told you in the previous video. Now this going to the third step, which is the elongation step. In the elongation step, the DNA polymerase will start copying the DNA sequence, and to give the copied DNA. Now we're interested here, so we're not interested in this strand anymore. We're interested in this strand. So when the DNA polymerase start to copy the DNA sequence, when it reach the probe, it will start to degrade the probe. Okay, so it will start to degrade the probe. Now, when the fluorophore is degraded from the probe, and so it's not close anymore to the Kencher, and so the Kencher cannot absorb the fluorescence coming of the fluorophore anymore, at this point, we can see the fluorescence coming out of the fluorophore, because the Kencher cannot inhibit the fluorophore anymore. The Kencher cannot absorb the light coming out of the fluorophore anymore. So the polymerase will continue by copying the DNA and by degrading the probe. Okay, like this, like as you are seeing here. So the probe will be completely degraded when the polymerase is copying the parent DNA. So let's go back here. So here, when the probe is degraded, we can see the fluorescence coming out of the fluorophore. Okay, then the polymer will continue copying the parent DNA. Now, how does this work? Now, when we have, going back to the normal PCR, when we have the PCR, then we will get more and more sequences at each cycle. So let's say we have Tachman in the medium. So in each cycle, there will be more DNA sequences and more DNA strands, and then more probes are binding to the DNA, and more probes are degraded each time. So at each time, so at first, let's say we will get a little bit of fluorescence, then we will get more and more and more fluorescence, and this what can be detected by the spectrofluorophore. We'll see spectrofluorometer we'll see in here. Now let's speak about a little bit about the advantages and disadvantages of the specific detectors, or the Tachman. Of course, the first advantage is the specificity. It's specific. It's specific to a certain sequence or to a certain gene. And because of this, I can do the multiplex real-time PCR. What is the multiplex real-time PCR? It's to perform real-time PCR to many genes in one tube. And yeah, I can do this with the specific detector, because let's say we have gene 1 and gene 2. We can use two different specific detectors. The first is coupled with the blue fluorophore. The second is coupled with the red fluorophore. We can also use green fluorophore. We can use multiple specific detectors, or multiple Tachmans. And then this will be very specific to G1. This is specific to G2. And then if I have one DNA sample and I want to perform several real-time PCR in one tube, I can, for several genes, I can use the specific detector. Some of the disadvantages, it's hard to sequence because it needs to be highly specifically sequenced. So it's expensive. And there is a probability to false results, which is derived sometimes from the inability of the Kencher to totally inhibit the fluorescence coming out of the fluorophore. Sometimes the Kencher will be not so effective in inhibiting the fluorophore, which might give false results, but this is rare. There is very important application in the real-time PCR is the reverse transcription. it's not an application. Actually, it's not an application. It's reverse transcription is highly used with real-time PCR. So let's say we have RNA virus, or we have a human cell, and we need to measure the expression of a certain gene. We need to measure how much this gene is expressed. Why? Because some genes, the expression of some genes differs between normal cells and cancer cells, for example. So the measuring or the analysis of gene expression is highly used in cancer cells. In this case, we can in this case we take the messenger RNA sample out of the cell, or extract the messenger RNA extract out of the cell, and we undergo something called reverse transcription to produce the complementary DNA out of the messenger RNA. Now I'm going to speak really in general about reverse transcription. So in order to perform reverse transcription, we need the parent messenger RNA. We need also the primer. We need the DNA contents to produce DNA. We need a very specific enzyme called the reverse transcriptase, which is used for reverse transcription. We need also RNAs, and we need DNA polymerase. Now, how reverse transcription is done? It's like this. So we have messenger RNA or RNA sample. So the reverse transcriptase copy the messenger RNA. It copied the messenger RNA. It produced DNA from RNA. So it's not transcription. In the normal transcription, we copy DNA to RNA. In the reverse transcription, we go back. So we copy DNA from RNA. Okay, so we copy the messenger RNA, we get something we call the cyclic DNA. After this, the RNAs will degrade the messenger RNA, okay, and then the DNA polymerase will copy the first strand of DNA, and then I will get double-stranded DNA from the RNA. I can use this double-stranded DNA in the PCR, in the real-time PCR. This is highly applicable in real-time PCR, actually, as I told you, in cancer, in the diagnosis of HIV or other RNA viruses. It's highly applicable. Now you know that in real-time PCR, the spectrofluorimeter measures the amount of DNA in the sample continuously during the PCR process. But how can I get the data? Or yeah, the amount, how can I get the data? So the quantitative real-time PCR, of course, real-time PCR is quantitative. So this is the machine, let's say, of real-time PCR. Normally it's engaged with a computer program, and which gives me the data in this form. Like, this is what we call the amplification curve, or in this form, or in this form. Anyway, it's the same. We have the same elements. So this is the fluorescence. What we see here in the y-axis is the fluorescence. In the x-axis are the cycles of PCR. Here we have 38 cycles, for example, 40 cycles. It depends. And this is the amplification curve. So you're seeing multiple curves because, as I told you, we can apply real-time PCR for several samples at one time. We can apply it in the 96 Wells plates. And so we get several amplification curves. You are seeing here three overlapping curves, because sometimes we apply the same samples 3 or 4 times, like in a triplicate, to ensure about the results. So this is what you're seeing here, or this is what you're seeing here: multiple amplification curves overlapping over each other, because they are amplification curves for the same sample. So we apply the same sample in several tubes to ensure about the result. Now, in all the curves you're seeing here, we have general elements. And the elements are, so this is the fluorescence, as I told you, this is the cycles. And this is very important. It's called the threshold line, what you're seeing here, or here, or here, the threshold line. And this is the most important value because it's the CQ value or the CT value. It's the threshold cycle. It's called the threshold cycle. And the threshold cycle, it's the cycle at which the amplification curve intercept with the threshold line. So the intersection between the amplification curve and the threshold line. It's called the CQ or CT, the threshold cycle. Why it's so important? Because if you see here, going back to here, for example, the CT here is somehow 22 at cycle number 22, but here it's at cycle 31. So let's go to here. What you're seeing here is that the fluorescence coming out of the sample at first is too minimal, and then as PCR goes on, it starts to increase. So when we have higher amount of DNA at the beginning, the amplification curve will go faster because we have higher amount of DNA from the beginning. And when we have very little amount of DNA from the beginning, the amplification curve will start to increase in a delay. So the curve which cross the threshold line first is the sample which had the higher amount of DNA at the beginning of the PCR, and the sample which crossed the line lately is the sample which had the less or the least amount of DNA at the beginning of the PCR. So we use this to compare between samples. So if I have several samples, I can use real-time PCR. Then I can know from the CT which sample had the highest amount of DNA at the beginning. So this is very important value, the CQ or the CT, and you can see here after several or certain cycles, the curve becomes non-exponential. Here we have the plateau phase because after several cycles we don't get any increase in the amount of DNA anymore. Maybe because the polymerase become inactive after a certain time, or because because the polymerase becomes inactive after certain time. And here is the exponential phase in which the amount of DNA increases exponentially. So this is everything I wanted to tell you about real-time PCR. Yeah, if you enjoyed the video, don't forget to like, don't forget to share it with your friends. And if you have any questions or any question regarding real-time PCR, don't forget to leave your question in the comments because I will answer you. If you enjoy my videos, don't forget to subscribe my channel. There will be many other interesting videos. So yeah, so subscribe my channel, like the video, leave your comments, and see you in the next video. Bye.